With today's advanced sequencing technologies and laboratory techniques, scientists are able to get detailed snapshots of activities in the cell nucleus, in bulk tissues, or in single cells. For example, we can measure the gene expression levels of a cell and identify variation in the number of transcripts produced for each gene. Or we can look at where transcription factors bind. Functional genomics assays such as these produce data, which are tied to specific individuals. While we can sequence the DNA of a sample once, there are several different functional genomics assays that can be done in tandem. These data have extremely high utility to the human genomics research community. However, they also present a significant privacy risk. Many functional genomics data types contain variant information about individuals, which is often superfluous to the biological question at hand, but is still present in the data. They are also inherently tied to the biological question. For example, changes in expression of genes in patients with a psychiatric disease or HIV are typically assayed with functional genomics experiments. In the wrong hands, this variant information can be used to reveal private, stigmatizing phenotypic information about study participants, such as disease status. Because of this, high-utility functional genomics data have had to be kept behind controlled access to prevent bad actors from misuse. However, this controlled access storage also makes it more difficult for scientists to access the data and use it to answer important biological questions. To solve this problem, we designed a data sanitization method to maximize data utility while providing privacy. To achieve this, we take the original data and split it up, putting the sensitive information, which is typically not needed to study genome function, behind controlled access where it is protected from bad actors, and keeping the rest of the information in a sanitized form, open access, for scientists to use readily. The sanitized open access data still contains high utility information, such as gene expression levels, but has simply been sanitized of all variant information. In this way, we can make functional genomics data accessible to the research community while protecting the privacy of study participants.